The native wildlife of Australia is crucial to our country. It is important to our environment, important to our history, and important to our identity. Therefore, it is crucial that we do what we can to protect our native animals. Since the arrival and settlement of Europeans in Australia, the bandicoot population has significantly declined, and as a result, they are now classified as endangered species and listed on the Threatened Species Act 1995. There are 3 species of bandicoots: the southern brown bandicoot, the northern brown bandicoot, and the long-nosed bandicoot, with 2 populations located around northern Sydney. There had previously been confusion over the identity of the species of bandicoots within the Sydney region, as both the northern and southern brown bandicoots are very similar in appearance. This made it hard to confirm whether the Sydney populations were of sympatric origin or in fact the same species. A DNA test to confirm the identity is therefore extremely important, as previous distinctions based on morphology, such as the biological species concept, are not accurate enough to distinguish between the 2. Since the use of DNA has become available in the veterinary field, it has provided countless advantages to many aspects of animal health and welfare. DNA can help to provide information about the distribution and behavioural patterns of species, and is crucial for breeding plans and conservation management. Other advantages include the use of DNA to diagnose various diseases and whether certain genotypes will increase the risk of susceptibility to a particular disease. To be able to make smart and efficient choices when breeding animals, to maintain diversity among species as well as maintaining a healthy population, and to identify an animal's lineage as well as avoiding inbreeding and problems associated with this.
In order to correctly identify the different sympatric species of bandicoot, a DNA test known as polymerase chain reaction restriction fragment length polymorphism was used.
PCR was used for DNA amplification in which multiple copies of a specific gene sequence are produced. For this test, mitochondrial DNA at the cytochrome B region was amplified as it evolves rapidly and can be used to identify relationships among recently evolved species.
The initial DNA sample was extracted using the salting method on tissue removed from the ear of the bandicoot. This process involves crushing the tissue and incubating it in a sodium chloride solution to allow the DNA to precipitate out. The mixture is then shaken, centrifuged, decanted, and washed in ethanol.
The PCR process begins by mixing the extracted mitochondrial DNA with primers, Taq polymerase, nucleotides, and buffer. The mixture is then heated to 94 degrees Celsius in a process known as denaturation, in which the DNA helix is forced to separate. Annealing occurs as the mixture is cooled to 60 degrees Celsius, allowing primers to bind to the end of the target DNA sequence. Finally, extension occurs at 72 degrees Celsius, the optimal temperature for Taq polymerase to join the nucleotides to the 3' end of the primer.
Taq polymerase is used during PCR because it is highly thermostable and can thus cope with the high temperatures required. It takes 3 cycles to produce the first 2 strands of target DNA. After 3 cycles, the number of target DNA strands produced increases at an exponential rate for a further 35 cycles.
RFLP utilizes differences in homologous DNA sequences to identify differences in individuals. Different species of bandicoot have slightly different variations in their genetic sequence for the cytochrome B gene, which can be observed in RFLP.
The first step of RFLP is enzyme restriction, in which a restriction enzyme is mixed with target DNA. The enzyme then cuts the target DNA into fragments at specific nucleotide sequences that are unique to the enzyme. This test used 2 enzymes, MSEL and SSPL.
The next stage of RFLP is electrophoresis on a 4% agarose gel. Electrophoresis is a method of separation, causing the fragmented DNA to spread out and form unique bands that reflect the size of the fragment of DNA. Once the DNA is loaded into the gel and an electric field applied, the DNA fragments are pulled through the gel at varying distances.
This event occurs due to the negatively charged oxygen atoms on the phosphate backbone within DNA. When the gel is placed in a solution able to conduct electricity, the DNA fragments are attracted to the positive terminal due to their negative charge. The DNA movement through the gel is called reptation. Smaller DNA fragments migrate further and faster as they can fit more easily through the pores of the gel than longer fragments. This phenomenon is called sieving.
The gel has two central properties. Primarily, the gel acts to slow the passage of the DNA fragments along the electric field. The gel also suppresses the thermal convection caused by the electric field, protecting the DNA from distortion. Once the electric field ends, a tracking dye, ethidium bromide, stains the DNA. Ethidium bromide binds tightly to the DNA double helix structure and glows when UV light is present. The pattern of bands formed are unique to each species of bandicoot. Image A shows the electrophoresis results from the restriction enzyme MSCL on both male and female bandicoots. The two distinct bands in the macroorus are distinctly dissimilar to the three bands of the nasuta and the single obliceous band. This can also be seen in image B for the digestion enzyme SSPL. Whilst the northern and southern brown bandicoots were unable to be distinguished by the MSCL restriction enzyme, the correct identification was obtained using SSPL. These results are significant in their ability to correctly identify and distinguish between all three bandicoot species. This is a more practical and accurate method of species identification than previously available and has practical significance in the field of conservation, breeding programs and disease management.
RFLP-PCR is extremely accurate at detecting single nucleotide changes in DNA between organisms. An alternative PCR method is nested PCR, which differs from RFLP by using two different primers when replicating DNA as opposed to the one primer used in RFLP. This reduces the chance of copying the incorrect section of DNA or for the DNA to be contaminated. Whilst RFLP PCR is slower when distinguishing between different organisms, it is more accurate than nested PCR in identifying differences between species. ARMS-PCR, or Amplification Refractory Mutation System PCR, is used to detect single-base changes in DNA. ARMS-PCR uses one primer that can't replicate mutated DNA and another mutated primer unable to replicate normal DNA in order to distinguish between organisms. It is more accurate than RFLP when only a single change in nucleotides constitutes a different species.
The DNA test of utilising an organism's mitochondrial DNA for DNA barcoding is an extremely powerful tool for identifying and contrasting species and their lineages. Before DNA testing, methods such as crossing two individuals to see if they are reproductively isolated, or examining morphological similarities between individuals were used to classify species. However, both these methods are slow, expensive, and sometimes inadequate in properly identifying a species. For example, in the case of our journal article, the bandicoots cannot be distinguished accurately on morphology alone, and attempting to breed wildlife would be time-consuming and a financial risk, with no guarantee of being successful. It is much more efficient to isolate and amplify a specific DNA sequence and compare this sequence between animals. This is a rapid and objective way to allocate animals to a species. Identifying endangered wildlife through their DNA is especially important in order to understand reasons why the species population is declining and implement preservation strategies in response. This may be through understanding their geographical distribution or even recognising a mutation in the DNA itself, which may cause disease to the animal. DNA sequences can be compared across species to see if some are more prone to the disease than others and gather information about the disease, such as if it is heritable. DNA sequencing is especially important as it can be applied to all phyla, allowing an evolutionary tree to be formed based on similarities in base sequences. This provides a way to build on our knowledge of the reasons behind animal physiology and ultimately the history of life on Earth.